give this talk, it's actually humiliating to talk about a multi-zillion-dollar therapy that is currently being offered to a few following Dr. Faulkner's talk. And I think my hope, and maybe during the question and answer session, that those of you who are the up and coming generation will struggle with some of these issues, because I think it's really coming to immense proportions. And we were talking earlier, we're doing our first gene therapy patient for thalassemia. $5 million is the cost. And we have a team of 1 million. There's literally, for every patient, there's like 20 people I'm tripping over to try to do it. And so there has to be some balance. That said, I'm hoping that some of these therapies can actually advance the science that then ultimately, like computers, I keep hoping that ultimately over years will become more affordable and really sort of move the field forward for salvaging our kids. But that remains to be seen. So I'm going to talk about chimeric antigen receptor T cell therapy today. No conflicts to disclose. And I'm going to go pretty quickly because I speak quickly, and then we can have questions at the end. So CAR T cells are exciting because it really is a new therapeutic paradigm. And what are they? Well, I always ask the residents, what's a chimera? Nobody ever knows. It's written up on our chalkboard in our BMT unit, but it's two things smushed together that don't really belong together. And if you can remember that, you'll understand the pathophysiology of CAR T cells, and then we can talk about the toxicity and efficacy briefly. So in classic, I think, Greek mythology, it was a lion and a goat, but it can be sort of anything. So what is the concept? So you take patient T lymphocytes and this is for Kymriah CAR T cells, the most common way now. You take the patient's T lymphocytes that are collected by pheresis, then you genetically engineer them. So CAR T cells are gene therapy, basically, and we use a viral vector. Currently, a retroviral vector is being used. I just highlight that because patients will be HIV positive because they're picking up the HIV genome, even though they're not actually HIV infected, of course, for months after CAR T cell therapy. And then what you end up with is a chimera. And this is the most important thing right here. And this was the brilliant idea of Carl June, who presumably will win a Nobel Prize for this, coming from CHOP and some of his clinical cronies that did it with him. So you have antibody recognition, and you're linking antibodies to T cell expansion and cytotoxicity. So they took the two different ways that a lymphocyte works, antibody recognition and T cell receptor recognition and expansion, and link them together. And that is really brilliant and a new paradigm. So it uses the native antibody recognition, and then it uses the T cell signaling, and it makes it a living drug, which is a little nauseating to say, but it expands dramatically after infusion. So when you give regular chemotherapy, you give a dose of chemotherapy, and that's that. This you give, and these T cells, if they work, will expand logarithmically during the days after T cell infusion, after CAR T infusion. And that's it doing its work. Someone else made this slide for me. So the product that we use the most for CAR Ts is tisagenlecleucel. And if you can say that after this talk, then that's better than most people. Why they named it that, I have no idea, but the trade name is Kymriah. And that's a T cell activation domain coupled to anti-CD19. So each CAR T cell product currently is just specific for the one thing the antibody attaches to. And then there's things in between that allow the T cell expansion, and the 4-1BB co-stimulatory domain, at least to date, has been shown to be sort of the most reliable one, and is the one that's FDA approved for Kymriah. And then you have the signaling domain that drives the T cells to expand. So how do you make them? It's a multi-step process. We've done this now for probably 5 or 6 years and learned a lot. One, it's very interdisciplinary. So you need the leukemia team, you need the gene therapy team, you need stem cell transplant, apheresis, and cell processing units. We, in most places, own the infusion of the CAR T cells on our service because we're fact regulated, and any cell product has to be given by stem cell transplant regulated people. If you're coming up with new ways of doing it, you could imagine doing it in all sorts of different ways. But basically, you collect the patient's T cells, you transfect them with the retroviral vector, and then you give, it's been shown, I'm not going into the incredible details because you won't remember them probably, but you give lymphodepleting therapy to make more room for the T cells to grow, which has been shown to be Flu-Cy is the one that is most commonly used. That can be given as an outpatient, and then you bring them in, you infuse the CAR T cell product, and you monitor the patient going forward. And that's what it looks like. And it's dosed, it's a drug, it's dosed per kilogram weight, and that is the product that you get when you take these cells and then ship them off to Novartis, which is the drug company that makes them, and then get the product back to use. Good to remember there's a time lag in there, and for all gene therapy, so brief detour, in the old days, all of gene therapy was only done at major academic centers who had cell processing labs, and they were like, we're the only people in the world that can do this. What Kymriah showed as proof of principle is that actually you can ship these things and have a drug company make them. And that comes with benefits and disadvantages linked to them, but definitely there's a time lag because you have to ship the product, they make it, they check that it's okay, and then they ship the product back to you, you receive it, and then you give it to the patient. So what were CAR T cells made to tackle? Well, as I told you, Kymriah has an anti-CD19 antibody, so it can target anything that expresses CD19 preferentially. So what the indication is for relapse refractory CD19 pre-B cell ALL. Luckily for the world, that's a pretty common pediatric childhood malignancy, although still relatively uncommon if you're talking about thalassemia and other worldwide problems. And I think it's important to remember that. And what they found is that there are these refractory kids with CD19 pre-B ALL, and that this product could hopefully get them into remission. And then we'll talk a little bit later, and either be a definitive therapy or be a bridge to a definitive therapy of allogeneic stem cell transplant. What's being studied now is using it for upfront therapy for ALL, and the hope is that it will decrease relapse in very high-risk patients, decrease long-term toxicity, and decrease resources and length of cost of therapy. So what I've learned watching this over the years, whenever you offer a really, really, really expensive product, you always tell how it will cost down the road. That is often true, but you still have to pay the upfront cost in order to get that cost savings down the road. And that's something that people are struggling with. Right. So Kymriah, again, the FDA approved product, the most important thing you would want to ask is how effective is it? And there's been a series of clinical trials in support of Kymriah, and the pivotal trial was actually called the Ileana trial. And the thing about this slide, it will show that it now started in 2012, so there's been relatively long-term follow-up. But what gets lost in this a little bit is the ends. So, there were 60 patients in the pivotal Ileana trial, 29 in the supportive trial, 59 in the supportive CAR T cell trial. So it's still a fairly small number of patients. If you ask how many patients have received a matched sibling donor transplant for thalassemia, thousands, truly, right? Probably many, many thousands. It started in Italy in the 80s, I think. And these patients have been followed for 30 years. So the data appears to all say the same thing, more or less, but it's still a relatively small number of patients. And what they saw was that the duration of remission was consistent across the trials. And even with longer term follow-up, the curves appear to plateau, and that they end up plateauing somewhere around 50+% for maintaining remission with Kymriah products. And there's a new study that just came out by Bader and Blood in 2022, and this was from Germany, very interesting, and it looked at people who relapsed after transplant and then went on to get CAR T cell therapy. So you had high-risk ALL, got an allogeneic stem cell transplant, and then relapsed. And again, their survival appeared to be about 50%. And again, the question is always, is this definitive therapy, or is this therapy that is going to be a bridge to yet a second transplant or a third transplant for some of these kids? But I would say when I talk to the residents, we play games on rounds, and one game is, what do you know for sure in oncology or the world, whatever? So one thing we know for sure is if you relapse early with ALL, that's worse, right? So every study shows that. I would say we're getting to know for sure pretty well that at least the way we give it now, about half of children will have a long-term disease control with Kymriah therapy. What are the toxicities? Well, they're getting less. In the very beginning, people thought that that having lots of disease would make CAR T cells more effective, sort of like rituximab. If you put it in when there was a lot of leukemia, the T cells would expand more. So it was given to patients who had, many, many, many circulating blasts. That has turned out not to be true, and now the patients end up having better disease control before transplant, and so the toxicity, and I think we're just better at it. So now we very rarely send somebody to the ICU. Basically, if you can remember this slide, this sums up all the toxicities quite well. The biggest one we're going to talk about is cytokine release syndrome, which is a from expanding T cells that you would see in other settings as well. And then the neurologic toxicity, which is called ICANS. And then the other things that we think about are insertional oncogenesis, because this is gene therapy and it's inserting into the T cell genome that has not been seen yet. And as I said, this started in 2012, unlike, I could talk forever about sickle cell leukemia after gene therapy, which is an ongoing problem, but this does not seem to be a problem yet. And then there's, you could get anaphylaxis and drug reactions, and then there's on-target off-tumor toxicity, which is B-cell aplasia. So you're attacking CD19 positive cells, as you would expect. Those are your normal B cells as well. So cytokine release syndrome is basically a systemic inflammatory response. It's an expected on-target toxicity. It ranges from mild to terrible, with needing pressors, needing oxygen. And in the early days, people dying. That has become much less now that we have, we're taking people mostly with a lower tumor burden. This is another weird paradigm. It's FDA approved, but you have to be vetted as a center to give it, and you do have to report your data. And one of the things you need to do, sort of like a research trial, only it's not a research trial, is grade your CRS and then report it to the CIBMTR. And this is just basically goes from fever and some constitutional symptoms up through these grades that have now been, it's a formalized grading system up to grade 4 CRS. For grade 4 CRS, the treatment is tocilizumab. I don't know if anybody knows what it is. It's an anti-IL-6 antibody. And what Steve Grupp, who was the clinical component of Carl June, said is basically the first child who's still alive now, 10 years after, just 10 years after CAR T therapy for refractory ALL. They were staying in the ICU. She was having horrible CRS, and the resident was standing there and said, I don't know, we used tocilizumab in someone with CRS with rheumatoid arthritis. He was like, okay, tocilizumab, and there we have it. And so other things may work better, anakinra, any of the other biologics, or something else completely, but now it's the one that's approved, and nobody's going to go back and do those studies. It is $1 zillion, is one of the things about tocilizumab to know. And then this is the sort of the percentages of people who get it. It's going down. Now we see probably about 10% of people who get grade 3 or grade 4 CRS. The neurotoxicity is interesting, also is getting less and getting less persistent. Grade 1 is malconfusion. Grade 4 is life-threatening confusion, somnolence, cerebral edema. We started our CAR T journey using a product that Sloan made that ended up being the loser instead of Kymriah. And there was a patient at the Brigham who was also using the same product who had a headache, taken to MRI. The MRI was read as okay, and he herniated in the elevator back up from the MRI and died. And so it was horrible neurotoxicity. Kymriah had less neurotoxicity all along, and now we're sort of treating it less. The thing to know about neurotoxicity is that steroids are the treatment more than tocilizumab. People don't really know if tocilizumab would help. Often it can overlap with CRS, and you get both, but steroids are the drug of choice. And just a brief aside, and learned from Egypt and Alaa al-Haddad, the transplanter there. So the early theory was that you never wanted to give these drugs, toci or steroids, unless the patient was an extremis, because you would abrogate the T cell response. It turns out that's probably not true, and giving low doses of steroids early on in places that don't have tocilizumab, and this comes up not with CAR T cells, but with haplotransplants as well, is probably just as efficacious, and you don't need to sort of drive people to extremis in order to have effect. And this is the things that people see and the percent that they see them in. This was a young boy that we treated. And so his manifestation of ICANS was a facial droop, a cranial nerve 7. He got an LP, it was negative. He got steroids, and he completely recovered, and also is a long-term survivor, having relapsed early after transplant. Briefly, you can see prolonged cytopenias. It's pretty rare, somewhere around 10 or 20% total cytopenias. And you try growth factors and you do bone marrows and make sure that they haven't relapsed. And most of these people will recover on their own, although there's reports of needing a stem cell boost. Hypogam, you expect to see, right? Because you're giving anti-CD19, and that's what's on mature B cells. And basically now the summary of all of these slides is that people watch you for hypogam, and the way to know when your T cells, your CAR T cells are dying, they've stopped expanding and are dying off, is when you begin to make your own IVIG. But during the time that they're working, you need monthly IVIG, which also adds to the cost. So how do we know if CAR T cells worked? Well, you get a bone marrow biopsy and aspirate at day 30 and you document response. And one thing when I talk to presidents and fellows about Kymriah that is brand new, it's the only chemotherapy I know about or drug where if it didn't work, you get your money back. So it's $500,000. And if your day 30 or up to day 44 marrow shows that you don't have remission, Kymriah, Novartis refunds your money. So what does that tell you? It tells you everybody relapses after day 44. That's not where they would have set the bar, right? They would have set it at day 60. Day 45, you get nothing. But it is interesting, and you must look then to document response. And insurance companies are very interested in us documenting response. And if not, you get your money back. So that 50% curve isn't the 50% who get their money back. This is just people who don't have an early response. How do you fail? Why do you fail? You can fail because the CAR Ts aren't active enough. They're not wide awake enough because you've gotten tons of chemotherapy or whatever. It's not persistent enough. There's sanctuary sites, works well in the CNS, may not work as well in kidneys and things like that. And then what people worry a lot about is clonal escape with CD19 negative cells. So is CAR T therapy definitive? Well, nobody knows. Approach A is that you hope it's definitive and you just watch. And if you lose B cell aplasia or you do multiple bone marrows, you see MRD coming back, then you intervene. And approach B is that you go straight to transplant saying, oh my goodness, I've gotten remission. I'm going to consolidate that. And I was in Berlin like probably 5 years ago now. And the talk was pro-con, can CAR T cells replace allotransplant and ALL? And this was a slide that I think I should attribute. I can't remember who said it, but who knows? We know how allografting works. We don't really know the long-term persistence of CAR T cells, and every center has a slightly different approach. What we do is if you haven't had a transplant, we go to transplant. If you have had a transplant, the toxicity is much greater, and we watch you. Just going to skip. So other novel aspects of Kymriah therapy. It's the first FDA-approved gene therapy. Now we have two more. I was talking to Dr. Faulkner. We have thalassemia, and we have adrenoleukodystrophy, and a billion more for hemophilia and not bone marrow diseases. So this is coming. So it's not on a research trial. It's more generally available, but safety data is hard to capture because what makes a research trial partly is being mandated to report data and being mandated to have a budget that has all the little people running around reporting the data for you. And it's centrally manufactured by a for-profit company, so you don't need this multi-million-dollar GMP facility to deliver cutting-edge therapy. And that's good. It opens up from an equity standpoint. So in summary, this is really a new paradigm of therapy for difficult ALL. At this moment, it has to be CD19 positive, and you have to document that before you can get Kymriah. It's FDA approved and technology driven, so access will rapidly expand, but data will diminish. So, 20 years ago, something like Kymriah, we would be the only pediatric center in New England delivering Kymriah level therapy. But now, if you're a transplant center and you get vetted with basically a multiple choice test after watching a slide deck, then you too can give Kymriah. It's unique toxicities that appear amenable to our supportive care. The efficacy is real, absolutely, but incompletely understood. And the cost issues must be addressed to ensure equitable access. $500,000 is what is the product cost. We charge, I think, $1 million for the whole package because you have to get pherese and lymphodepletion therapy. So the $500,000 just gets your cells made and shipped back to you. And hopefully that will go down in time. It has not yet happened. And then I think the other take home is that the generalizability to other leukemias and solid tumors are unclear. For those of you in training, solid tumors express a billion different antigens and they change rapidly. So there are trials undergoing, but whether this will ever be good for osteosarcoma or Ewing sarcoma is really a question. And as you can imagine, for AML, you could have an anti-CD33 CAR T cell, but then you kill all your bone marrow. Those are your stem cells, and then you're completely aplastic, and you have to be rescued by an allogeneic transplant by definition, or the CAR T cells don't work. Also, the early efficacy data isn't as good for AML. So it may be like Gleevec. It may be that pre-B ALL expressing CD19 was the perfect magic match for CAR T cells in leukemia, or the technology will change and it will have wider applicability. And I think I can end there, actually. Yes, thank you.